It's my very great pleasure to introduce to you today's speaker, Dr. Ivo Mueller. For those of you who don't know, Ivo is a new lab head appointee at Mueller I as of this year. Ivo did his PhD at the Swiss Tropical Institute up to year 2000, and then did his first postdoc in Queensland, in Brisbane, attached to UQ, but on secondment to the Papua New Guinea Institute of Medical Research. And subsequently, in 2002, he moved to the PNG Institute of Medical Research as head of the Malaria epidemiology unit there, and then rose to become head of the Division of Vector-borne Diseases and ultimately deputy director for research at the Papua New Guinea Institute of Medical Research. I met Ivo up there, and myself and a lot of people in the malaria community in Melbourne and throughout Australia, and indeed throughout the world, have been fortunate to collaborate with Ivo in his studies on the Southwest Pacific. Quite a few of you are in the room. And we're absolutely delighted that Ivo agreed to join us at the beginning of this year in his new position. So Ivo's going to give us background on some of the more important, or some aspects, just a small aspect, a small slice of the very interesting.
Thank you very much, Louis. So this talk is really going to be, even though I do consider myself a biologist, this talk is going to be very much epidemiology. So no gels and sequences and fax profiles and other kinds of things. It's going to be, in that sense, really highlighting the background of what we've been doing over the last 10 years or so, and in that sense, putting into the epidemiological context some of the more in-depth biology studies that we are doing, or I'm doing in collaboration with other people here at WEHI in the division, but also elsewhere in the world. Also, I'm really going to focus here on the comparison of the two most common human Plasmodium parasites, which is Plasmodium falciparum and Plasmodium vivax. And Plasmodium vivax is not an organism that has received much attention at WEHI, but I think actually most other places in Australia either, even though we are living here right in the center or right adjacent to the major area where that parasite exists. There is about 2.8 billion people at risk of vivax infections, of which about 90% live in the Asia Pacific. So we're really here right at a neighbor of the areas with the highest burden of that disease. And considering that it is supposed to cause somewhere between 80 and 300 million cases a year, it is actually quite an important parasite. But it has been severely understudied and also underfunded, the work for it, which is one of the things that I have been with some friends trying to change. So a lot of this talk really focuses on highlighting what are the difference between these two parasites and why is it actually interesting of not studying one or the other, but comparing them one to the other. Just to start off with a few, just one slide on the background of malaria and PNG. For those of you who are either not malaria people or haven't worked with PNG. So PNG, in terms of malaria, is the world in small. It goes all the range from highly holoendemic transmission to no transmission at all in the highlands. They're all 4 human Plasmodium species present. It's actually probably 5 because we have both form of Plasmodium ovale. Plasmodium vivax and also locally Plasmodium malariae can be very abundant. They can have, even Plasmodium malariae by PCR can have a prevalence rate that can be 15 to 20% of people in very highly endemic areas. Given the fact that you have high transmission and many species, you have very complex infections. Many people, about half of the people who are infected by PCR are infected with more than one species at the same time. So mixed species infections are very common. And there is really a very dynamic and changing relationship between these species if we look over the last 100 years or so in relationship with malaria control and also different drug treatments and changes that is. So this is a bit of the background. So PNG, because of its situation, is really ideally suited to start looking at the comparative biology and epidemiology of these two species, because we have a high transmission risk of both, so we can do intensive biology studies in a relatively small sample size. And we have started doing that, and I'm here going to present results of studies from the last 5 years that we have looked at comparatively for both species in 3 areas that I'm going to present. The first is the risk of severe disease. The second one is to look at age-specific risk of malaria and what that means in terms of patterns of acquisitions of immunity. And the last point I'm going to talk about are host genetic adaptations to malarial infections. Just quickly to highlight where the main differences are between the 2 parasites along its life cycle. If we have the mosquito biting here, the sporozoites going into the liver, that's where probably the biggest and most profound difference between the 2 parasites exists, and that's this stage here, which is the hypnozoite. The hypnozoite is a long-lasting, probably metabolically arrested stage in the liver that remains basically inert and then can reactivate after weeks and sometimes months or even years and re-establish blood stage infections without requiring a new mosquito stage infection. While in falciparum, all sporozoites go into the liver, they go through the merosomes and then release the merozoites and start the blood stage infections. Another major difference in the biology is that Plasmodium vivax will only invade very young red cells, reticulocytes, or even younger red cells, precursors in the bone marrow. And then that's another limitation that vivax has. I also would like to point out that that's not really different. It's more a qualitative difference that vivax is an extremely transmissible parasite. It does make gametocytes continuously when it has a blood stage infections. It makes them at very, very low density. And the vivax gametocytes are highly infectious. So we've known that for 50 years. When people fed mosquitoes on malaria therapy, patients that were several weeks blood slide negative for vivax, they still were able to infect mosquitoes. So it's a parasite that is, the vivax is a parasite that is extremely well adapted to the human host. It is a very efficient transmitter and also can sustain itself probably at very low transmission levels and in also small populations like residual populations and other areas. So there are some significant differences in the life cycle, and we're going to now look at some of the differences that does in terms of their epidemiology. So first to talk about the epidemiology of severe malaria, and there I have to start because vivax has been called in the literature in many textbooks as benign tertiary malaria. And because it is considered non-lethal. Although Bill Collins, who is one of the last people alive who did malaria therapy trials, will tell you, he once very nicely put it and said, vivax may not kill you, but if you have it, you wish you were dead, because it causes very, very high fever and the classical rigors and the malarial fevers and chills that are known. But the general perception is that it is benign and doesn't kill. It's even so far that a famous malariologist who works in Southeast Asia at a vaccine meeting that I attended actually went to the point of saying vivax does actually protect against severe falciparum illness and is therefore probably the best vaccine we currently have for falciparum malaria. So I'm trying to reflect on this a bit. So this is a classical spectrum of disease. Thank you. So that's a classical picture of what you see at the rural health center in Papua New Guinea in terms of patients coming in with fevers and how many of them have malaria. You see in very young children, and that's a misunderstanding many people have, in very young children, malaria is not the primary cause of disease. But it is the primary cause of disease. Between 60 to 70% of children between 2 and 10 years of age have a positive blood slide. The vast majority of them have a Plasmodium falciparum positive blood slide. The Plasmodium vivax, the highest proportion of Plasmodium vivax blood slides positive is actually in younger children than in falciparum. It's really in the 1 to 5-year-olds. The other species, they are present, but they're really not a big source of disease. So we looked at that health center, going through health center records and characterized which of these infections that come in do fulfill WHO criteria for severe malarial disease. And to our quite big surprise, we found that while, as expected, about 1 in 8 falciparum infections was associated with some with some form of severe disease, we found that actually close to 9% of vivax infections also were associated with severe illness. And contrary to what our colleague from Southeast Asia claimed, mixed infections, so if you had both falciparum and vivax, you had the highest risk of severe disease. When we look at the age structure of this, then we'll see that while young children have overall less positive blood slides, if they do have malarial infection, the risk that they do present with severe illness is clearly highest in children under 2 to 3 years. There are similar age patterns for both falciparum and vivax, so there's a bit less in vivax, but in generally the shape is different. And again, in all these younger children, we see that the mixed infections have by far the highest risk of causing severe disease. If we look at the clinical symptoms, and I have to stress here, this was done at an outpatient setting, so the clinical data here is relatively weak. But we can see that in vivax, we see a lot of respiratory distress, we see less anemia than in falciparum, and we see about the same amount of neurological signs, which means repeated fits, which means impaired consciousness. So overall, the clinical presentations at an outpatient setting between falciparum and vivax do not differ greatly. In the mixed infections, we had somewhat more anemia and a bit more respiratory distress, but again, the numbers here are so small that you can't really compare the mixed with the single infections. The next thing which we've done is, in a longitudinal cohort, I will talk a lot more about later, it's a cohort of children 1 to 4 years, we followed these kids very carefully for 16 months, and in this cohort we detected 30 cases of severe malaria. And because these were children that we followed up longitudinally, we were actually able to determine what is the risk of, prospectively, what is the risk of an infection leading to severe disease. And so what we see here is in falciparum, we had 4% of infections resulted in severe disease. We had about 1% of vivax infections that resulted in severe disease. And this is just mixed and everything else mixed up. So if we look at that in terms of the actual incidence rate per 1,000 person years, we have about 67 per 1,000 is the actual community-based incidence of severe disease for falciparum. The incidence of vivax is about 5 times less. So based on this, we can say that vivax is less often resulting into severe disease, but it is not a benign parasite because it can actually lead, when it does lead to severe disease, the severe disease is similar to falciparum. We did have 2 deaths in this cohort, but they were both due to Plasmodium falciparum. So we continued work at the major referral hospital in Madang on the north coast of Papua New Guinea, together with our colleagues from the University of Western Australia, Professor Tim Davis and Lawrence Manning. And we set up a cohort there that was a case control study that was for host genetics, and some of people at WEHI are working on these samples. Julia presented, I think 2 weeks ago, some data on immune responses in these children that relate to the same cohort. So what happens when you go at the hospital? There were 2,000 children that were admitted to the ward at Modilon Hospital in the time that we were monitoring. Of these, about 40% had fulfilled the clinical definition of having severe illness. Of those, about 40% were malaria cases in the site that they had a positive malaria blood slide. 13 we couldn't confirm by PCR, and they were excluded. And that's the spectrum that we had. So we had, most of these cases are Plasmodium falciparum, about 80% or so are falciparum malaria. But there is about 70% are falciparum malaria. There is about 10%, 8 to 10% that are severe vivax malaria. They are PCR positive only for vivax. They have no concurrent falciparum. And about 50 that were concurrent with vivax falciparum, and we had 1 single case that was concurrent vivax and malaria. But interestingly, the malaria probably did cause some disease because that child had a severe nephrotic syndrome that is that is known from Africa to be associated with chronic Plasmodium malariae infections. If we look at the clinical spectrum here of these cases, we see a big overlap of these different spectrums. We have severe anemia, which is about 1/4 of the cases, about 30% have impaired consciousness, and about 25% have acidosis, and acidosis tends to be the best predictor for mortality. But there are children that have different kinds of combination of these syndromes, as we have children that did not actually fall into any of these categories, but because they either had prostration or they had other symptoms that according to the official qualification qualify as severe disease, they were considered severe malaria cases as well. In terms of death, we had one death in a child.
Please note, the patient rest period commences in 10 minutes and will run from 1:30.
Okay. So we had 6 deaths. One was a PF case own. We had one death that was a child that died from cardiorespiratory arrest with a pure vivax infection, and we had 4 children that had mixed infections that died. So we had actually this is statistically significant that we have a higher mortality rate in mixed infections. So the conclusions from our work on severe disease would be that Plasmodium vivax is not benign. It does lead to severe disease and death, and that has been confirmed in studies from adults in India and Brazil in particularly. And however, when we're looking at the actual attributable risk, then Plasmodium vivax infections are indeed less likely to lead to severe disease. However, the mixed infections have the most severe symptomatology, and I do therefore think we can reject the idea that vivax is a vaccine for falciparum malaria. So that brings me then to the second topic, and that is a bit about the natural history of immune acquisition and age-specific risk of malaria. So the general understanding, very broadly, and that has been laid out much better by people like Kevin Marsh and others, is that immunity to malaria is acquired over a prolonged period of time. And interestingly, the first time I know that has actually been written like that was in 1900 by Robert Koch after he visited Papua New Guinea. He concluded that it took several years for people to become immune. He also concluded the last point here that there is hardly any cross-immunity between different species. So if you're immune against one species, that will not protect you against the other species of malaria. We have since learned that immunity is probably acquired in phases, that you first acquire immunity to severe disease, but you're still at high risk of uncomplicated disease. Then you start getting more immune against uncomplicated disease, and only at the very end, you start actually getting immunity against the risk of infection per se, and that immunity is always imperfect.
Attention, please. Can Kelly Braden please go to 1 West Orthopedics?
So the rate of this immune acquisition, we also have learned, depends obviously on transmission intensity, so that the more intense transmission is, the faster acquisition of immunity is. From earlier studies in PNG and other co-endemic areas, we also have seen that when we look at both parasites in parallel, we find that the prevalence of vivax tends to peak in younger age groups, that vivax illness is also commonly found in young ages, and that that's just what I've shown before, that we have a comparable symptomatology, at least, of severe illness, but it's higher for the mixed infections. So together, this would indicate that there is a faster acquisition of immunity to Plasmodium vivax, assuming comparable transmission intensities. And in PNG, the transmission intensity for both parasites tend to be roughly the same. So if we look at this now in some recent study, this is from a large cross-sectional survey of about 3,000 people in the CPIC area of Papua New Guinea, and both by light microscopy here and by PCR, we can see that in children under 3, Plasmodium vivax is the predominant infection. It has a higher prevalence than Plasmodium falciparum. But as we get into older age groups, the prevalence, in particularly by light microscopy of vivax, starts dropping quite dramatically, while really only in adults the prevalence of falciparum infections also starts dropping. But the cross-sectional studies don't actually allow us to tell very much, or the case control studies from the clinical things. What we really need to look at, we need to do longitudinal cohort studies where we can follow the individual child or the individual person over time and prospectively assess the risk of disease. They do allow an accurate estimation of the burden of infection and illness, and we can do also then these comparative studies of immune responses, but also because we have the parasites that infect the children, we can start in detail dissecting the interaction between the host and the parasite. So we've done a number of these things. The first one I'm going to talk about is a large cohort we've done in Madang between 2006 and 2010. It's actually a randomized clinical trial of an intervention called intermittent preventative treatment to prevent malaria in very young children. These children were followed up with active bleeds every 3 months, and there was a passive case detection where we just were seeing how often did they get sick and came to the health center. In total, we had 1,100 children, of which a third was in the control group, so they didn't get any drug. The other 2/3 got either artesunate and SP or arimidiaquine SP with the aim of preventing malarial disease. But I'm really going to concentrate to a large extent on this control group because these are the children that didn't get any drug and where we therefore see the natural risk of malaria in those very young age group in infants. So if we look in children between 3 and 15 months, we find that we have about 1 episode per child per year in terms of its risk, and 70% of those are Plasmodium vivax episodes. So more than 2 out of 3 malaria episodes that a very young child in Papua New Guinea experience are due to Plasmodium vivax. In somewhat older children, we still have roughly the same amount of malaria, but we start seeing more Plasmodium falciparum episodes, and the vivax episodes start going down a bit. If we are looking at prevalence of infection, I have to please note the different scale. This is basically 3 times the scale of here. We see that at any of these age groups, this is the overall prevalence here by PCR. At any age, we have between 2 and 5 times the prevalence of vivax infection in these young children as we have of falciparum infections. So clearly, in children under 21 months of age in Papua New Guinea, Plasmodium vivax is your predominant source of infection and disease. We then extended this into the older age group, and that's how I'm going through the cohorts. I'm going into older ages. This is a study which we've done in Eliza, also in the East CPIC here. It's a study where 264 children that were between 1 and 3 years of age were enrolled. We followed them up for 16 months. We seen them every 2 weeks to check them for the presence of clinical disease. Every 8 to 9 weeks, we took a blood sample from a finger prick. And in this study, and this I think is the only study that I know of in the world, we actually took 2 blood samples 24 hours apart. And I come back to why we're doing it. I also have to point out the excellent work done by the field teams in PNG because they collected 94% of all samples as per protocol. And that is a tremendous achievement in terms of both the children that participated and their parents and the field staff that they've done is. We don't often talk enough about them because that research without them is never possible. It's always a collaboration between scientists, field workers, and the community. So if we are looking at, again, first the prevalence of infection, you probably won't be surprised any longer by now that, again, Plasmodium vivax this is a PCR-based assay, where we see, again, Plasmodium vivax across the entire age spectrum has a higher prevalence, and it is really not varying very much. Actually, by light microscopy, it's not varying at all with age, while we do see an increasing prevalence of falciparum infection through that age group, as well as we're seeing an increasing prevalence of mixed infections. When we're looking at the incidence of clinical disease, we see very dramatically opposite patterns. In Plasmodium vivax, we see a significant reduction in the risk of clinical disease from children below 2 years, which have roughly 3 episodes per child per year, and by the ones that are above 3 years, where they have about 1.5. So there's about half the clinical disease in the older children than in the younger ones. In Plasmodium falciparum, we see the opposite. We actually see an increase in risk over time. And I'll explain afterwards why that is the case. If we are looking at the seasonality, at what happens in terms of season, we're also seeing interesting patterns. In Plasmodium, in the prevalence of infection, we actually see opposing seasonal trends. The gray area here is the rainy season. So as the rains stop, Plasmodium falciparum prevalence drops. The rains start, it goes up. The rains stop, it drops. Vivax, if anything, shows the opposite seasonal pattern in terms of prevalence of infections. If we, however, look at the incidence of clinical malaria, we see parallel seasonal trends. The reason for this, I think, is the fact that Plasmodium vivax can recolonize the bloodstream from long-lasting liver stages without needing mosquito transmission. But we also know, since immunity to vivax does seem to be acquired quite quickly, that it's probably these relapsing infections are less likely to be associated with clinical disease. So we may get a lot of infection coming out of the liver during the dry season, but they do not cause clinical disease. Therefore, they do not get treated, and the prevalence of infection stays high throughout the dry season. But because they do not cause clinical disease, the actual incidence of disease does decrease during the dry season. Transmission starts, new parasites are transmitted by the vectors by both falciparum and vivax, and we see a parallel increase in clinical disease as new parasites are being transmitted by the mosquito. So we weren't happy with just looking at blood slide and PCR, because we know that there's a lot going on under that surface when we all actually start looking at the genetic composition of infections. And in this study, as I said, we had the unique opportunity that we had 2 samples taken 24 hours apart. The reason we did that is because we know falciparum sequesters, and we therefore assume that in a single blood sample, we're not going to get the complete genetic composition of the infections at that time point. We thought for vivax we will get that, because vivax is not supposed to sequester. But we looked at both. So this is genotyping data. This is falciparum. This is vivax. What we'll see here is that, and the detectability is the probability that we find an individual clone in a single sample. So in falciparum, we have a chance of about 70 to 80% to find an individual clone in an individual sample. The ability to find that clone decreases as we have higher complex infections. When we have 3, 4, 5, 6 clones at the same time, that probability goes down. As it goes down, as we come to lower densities of infections. And that is not surprising, because these PCR methods, they do have sensitivity issues, and if there is and they also have issues of primer competition, that if you have many clones, you may not detect the minority clone always reliably. And that's the issue here with the multiplicity of infection. But also, if you have very low amount of DNA in your template in the first place, it becomes a stochastic thing whether you pick a clone or you don't pick a clone. Interestingly, firstly, these are 2 separate markers for vivax. Firstly, the detectability in vivax was actually worse, despite the fact that it did not sequester. We had more problem to detect an individual clone in a sample, and we find exactly the same but even stronger effect, that as densities become low, we have real problems detecting all vivax clones in a sample. So that does not mean that I'm claiming that vivax is sequestering, but it's clearly that there are significant issues in the detectability for both species. We then use this data to look at some molecular epidemiological characteristics. So I showed you before the prevalence in this cohort overall. The prevalence was about 30% for falciparum, and it was about 60% for Plasmodium vivax. The multiplicity infection is the average number of clones in a sample by each species. And for falciparum, it's about 1.5. So if a child is positive for falciparum, on average it has about 1.6 clones. It's twice as much for Plasmodium vivax. So vivax infections are significantly genetically more complex. If we look at the force of infection, and the force of infection is the number of genetically distinct clones acquired over 1 year at risk. And we're seeing again that vivax has roughly twice as many new clones that children in that age group acquire from Plasmodium vivax compared to Plasmodium falciparum. However, if we look at the total amount of clinical disease, it's roughly the same, slightly more Plasmodium falciparum disease than Plasmodium vivax disease. So again, that highlights they have a higher exposure, higher transmission, but they also have a faster acquisition of immunity. So the chance that a single one of these infection they acquire becomes clinical is significantly lower for falciparum than it is for Plasmodium, lower for vivax than for Plasmodium falciparum. Together with Terry Speed and Katie Benton, who is here, we've been doing some work to try to look a little bit what the relationship is between that measure of exposure, so the force of infection, FOI, and clinical disease. And these are some seasonal plots that we've done very recently, where you see for falciparum, we see a significant seasonality for both the clinical disease as well as transmission, as it varies with season. Interestingly, we have a shift here where the risk of disease peaks before transmission peaks. Now, this is pure speculation, but that could actually indicate what happens is that during the dry season, we lose some acquired immunity. So these initial infections, because this is basically where the rains start here. So these initial infections have a very high likelihood of becoming clinical. But as you go through the transmission season and children have had a number of new exposure, they do acquire a certain degree of unstable immunity, and therefore the actual per infection risk of having clinical disease tends to drop. In vivax, we see a very strong seasonality of the clinical disease, as we saw before, and a somewhat milder seasonal pattern in the number of new clones that are detected in the blood. If we now adjust for the seasonality in clinical disease for the differences in exposure, we find that there's a dramatic change in the seasonal pattern of falciparum. It actually goes to becoming non-significant. So this curve here does no longer statistically different from having no seasonality. So that force of infection really drives that seasonality. On the other hand, in vivax, we see a little bit of a change here in the dry season, but we see absolutely no difference in the seasonal pattern risk during the rainy season. If we then look at all different kinds of factors that explain risk of disease, we have, if this is basically predictors of clinical falciparum disease, we have very strong association with age. We saw that the risk goes up with age. We have a significant reduction, or that's the seasonality here, we have a significant reduction of risk with the use of insecticide-treated bed nets, so they prevent about 45% of disease. We also had less disease in children that had recently been treated for a malarial episode. If we now adjust this analysis for the number of new clones a child acquires, all these factors become non-significant. The only variable at all that explains differences in clinical disease in that age group is how many new clones are you acquiring. If we know how many clones a child acquires, we know exactly what the differences in risk are in that young cohort with limited immunity. We also explain the spatial variation that is significant here is completely gone. There are no spatial differences any longer as well in risk once we know the differences in exposure. Plasmodium vivax, on the other hand, we don't see any of that kind of relationship. Again, there is a very significant association between the number of new clones a child acquires and risk of clinical disease, but it makes no difference to the overall epidemiological pattern. The reason for this are two. The first reason is these children are much more immune to vivax than to falciparum. So differences in immunity here are overriding differences in exposure because everybody has acquired a significant amount of immunity by the time they reach 2 or 3 years of age. While here, immunity is very poorly formed, at least immunity against uncomplicated disease, so exposure is the dominating factor and it's not moderated by natural acquisition of immunity. The other reason here is that while force of infection, the number of new clone here really measures the number of new mosquito infections because each mosquito bite goes to the liver, comes out the bloodstream, gives you one infection. With vivax, on the other hand, we know that each mosquito bite can lead to several infections because it relapses out of the liver. So for vivax, the force of infection is a much less accurate measure of the actual exposure in terms of genetically completely distinct parasites. And I think these are the two reasons why we see these really dramatic differences in that association. The last study I'm going to talk about is a study that many of you here in Malaria who have heard different malarial talk would have heard about. That's the Mueller treatment reinfection study, which was a small study about 200 children from 5 to 13 years of age that we treated with antimalarials to remove all the pre-existing malarial infections, and we then followed them for 6 months and says to see when they got reinfected, and that's what we see here. Vivax again and falciparum here, and we see they get very quickly reinfected by 6 months. Basically, everybody had a positive PCR, and there is no difference whatsoever in the risk of reinfection, time to reinfection with the two parasites. They get roughly 5 new infections per child per year in that age group. This is a different population, but similar transmission dynamics. By light microscopy, we do see a little bit of different here at the rainy season, but overall, there is not much different either in time to reinfection by light microscopy. However, when we look at the risk of clinical disease, you will see that at that age, at 5 to 13 years of age, we only have falciparum disease. So the same number of new infections, a 21-time difference or 22-time difference in risk of clinical disease. Actually, we had more Plasmodium malaria infections in these children, clinical ones, than we have Plasmodium vivax. So by the age of 5 years, these children have acquired almost complete clinical immunity to vivax malaria, but they have significant risk of falciparum still remaining. However, acquisition of falciparum immunity is ongoing in these children, and we see this by comparing the risk of infections with different density between children that are above 9 and below 9 years of age. And we see that clearly they have acquired, the red ones are falciparum, the blue ones are vivax. They have acquired the ability to control high-density clinical infections and moderately high infection of Plasmodium falciparum as well. And that's why it turns out in the research we're doing together with people in Louis Scoville's lab and in James Beeson's group that has now moved to the Burnett, we are finding that this is an ideal group to look at correlates of protection of Plasmodium falciparum because they're exactly at that stage where they're going from at high risk to going to be at low risk. So the epidemiological evidence from this is that we can see in very young children about 70% are vivax, and vivax is clearly by far the most prevalent parasite. In the somewhat older children, we find that Plasmodium falciparum infections and disease increase, but the prevalence of vivax is constant, and the incidence of vivax decreases significantly. And in the older children, we find that there's an increasing ability to control high-density falciparum infection, but clinical disease is still quite common, while we basically have no longer any high-density vivax infections and no vivax disease. So I think we have hopefully quite convincingly shown that there really is a significantly faster acquisition of immunity to Plasmodium vivax, partly due to the fact that they do acquire significantly more clones of infection, probably due to the fact that the relapses from the liver keep boosting and contribute to that fast acquisition of immunity. This is not just a PNG phenomena, and it's not just a phenomena at very high transmission intensity. These are two other studies. That one's from Vanuatu, which shows in black vivax and in white falciparum. We show the same shift in the incidence of clinical malaria in much younger age groups in Vanuatu, where transmission intensity is much lower than in PNG. We find that even in Thailand, where transmission is really very low, and we find that even there, the vivax, which we see here in the reddish color and then the purple here, is the falciparum. We do see a significant shift of the vivax into younger age groups compared to falciparum. And that was even observed in the 1930s in Crete and on Puerto Rico, where it was well known that vivax was a disease of children and adolescents, and falciparum was a disease of adults. So I think this is a generalizable observation. Why? Why so much faster acquisition of immunity? I talked about the relapses, the fact that with a single mosquito bite, you do not get one blood stage exposure, but you get several blood stage exposure. And they can either be genetically identical, or if they come from a meiotic product in the mosquito, then they're at least 50% genetically identical across the whole genome. So you would probably get, this would result in very fast and efficient boosting of immune responses that are acquired with an initial infection. There are some discussions if there is less redundancy in key processes in vivax than in falciparum. And one of them that is always mentioned is the fact that the invasion of Plasmodium vivax into the red cell is it's not completely Duffy dependent. It has been shown since that vivax can invade Duffy negative red cell, but at a very, very low efficiency. So we have looked at the Duffy issue in PNG. We have looked at children that have acquired antibodies to block that Duffy binding protein interaction, and children who have a high ability to block that in an in vitro assay do indeed have significantly less malaria and also less density. We also have in PNG, we have the only other known population outside African populations that are Duffy, not negative, but at least Duffy heterozygote, so that have on one allele here, they have the same promoter mutation. That means that the Duffy is not expressed, and these children have roughly 50% Duffy on the red cell surface, and we see again a roughly 50% protection in these children against vivax malaria. So clearly that interaction with Duffy is crucial, and that may be one reason because there is not as much redundancy as in the EBA-RH system in falciparum, that it may take not as long to acquire that immunity. Having said that, very, very few children in nature do acquire effective blocking of that interaction. The parasite has evolved to safeguard that interaction very efficiently. The other reason is that vivax does not have the same kind of multi-gene family like PFE-MP1. So vivax is also not thought to sequester. Also, there's now evidence that it actually can bind to CSA and to hydrolonic acid. That the binding probably occurs rarely, but if it does bind, it can actually bind quite stably to tissue. So I think that's one that we will, in the next couple of years, have to rethink. How important it is, a different question. It can do it, but probably not very often. And now jump to the last topic, and that's the host genetic issue. Here again, the classic perception of Plasmodium vivax is that it's not lethal enough. It doesn't kill enough people to actually have a significant impact on the host genome. And therefore, people have classically interpreted the very different set of red blood cell polymorphisms that we find in the Pacific, which is really unique compared to Africa. They always have tried to link that to Plasmodium falciparum exposure because they just thought that the vivax is a benign parasite and therefore it can't cause enough selection pressure. One of these traits that is very unique to the Pacific is the Southeast Asian ovocytosis trait. It's caused by a 27 base pair deletion in the band 3 gene, or the human annular exchanger 1. It's lethal in its homozygous state. So we have never even seen a stillbirth that was homozygous for this deletion. However, we find it in extreme cases. In one population, we found a gene frequency of 35%. The normal gene frequency in coastal Papua New Guinea is around 10 to 12%. Considering this is homozygote lethal, there needs to be a pretty significant selection pressure to keep that mutation in the population at that frequency. Earlier studies that focused on falciparum have shown that it is associated with a complete protection against cerebral malaria. We have never observed a child with cerebral malaria that was an SAO genotype. However, we have cerebral malaria is not as common in PNG as in Africa, and is not as common a cause of death. So I'm not sure that is sufficient to keep SAO at that frequency. In addition, we do have some evidence that SAO may cause a higher risk of acidosis, and so it may actually balance itself. So we started to look at what is the effect of these human polymorphisms in our different cohort studies. So we're going back to some of the same studies again. So this is again the IPTI study with the incidence that you've seen before. We have genotyped all these 1,100 children in that cohort. We find 12% are SAO. We find that about 20% of them have alpha-thalassemia, and about 60% are heterozygote for that trait. We also do have some Gurbidge-negative children and some Gurbidge heterozygotes, but I'm only going to talk about the SAO here. So what happens in terms of clinical disease in these 1,100 kids? If we look at all episodes, we have a 31% protection against all forms of malaria. But this is purely due to a 40% protection against vivax malaria. We have no impact whatsoever by that trait on the incidence of falciparum malaria in these very young children, and they have really no immunity. So this is a pure genetic effect. It's not balanced by differences in acquisition of immunity. We've also looked at that effect in these older Mughal children, in the 5 to 10-year-olds. We couldn't look in the 1 to 5-year-olds because they live in an area where that polymorphism is not found. What we find there in that cohort, 13% of children are SAO. We find a 50% protection against risk of infection this time, not clinical disease. And I have to point out, it's much more difficult to find the protection against infection than against disease. So we find a 50% protection against the risk of reinfection by PCR as well as by light microscopy. Interestingly, we also found an effect against Plasmodium malariae, but we found absolutely nothing at all again in risk of falciparum infections. We then looked in these children trying to figure out a little bit of what may be the mechanism, and the one thing that we looked at, just one thing we looked at, is we started looking at if there was an interaction with the Duffy binding protein and the ability of the parasite to block that Duffy binding protein. The reason for this is that the Duffy binding protein is found on the red cell surface in a multi-protein complex that's formed by a dimeric band 3 molecule. And those dimeric band 3 molecules tend to become rarer on the surface of SAO cells. So most of the band 3 on the SAO red cell is in tetrameric or higher oligomeric form. And so we thought that may have an impact of that either their Duffy is not properly presented or it's not as much expressed on the surface. But what we found is that there isn't really a difference. So the expression is basically the same, and that had been shown before in another study, that here you could say it's just these 2 points that make the difference, but there is a second study that's larger that shows again that there's the same amount of expression on the surface. We then did use recombinant Duffy binding protein and had the Duffy binding protein bind to the same red cells from the same children, and then looked by fax, and we found absolutely no difference in the amount of binding of recombinant Duffy binding protein to SAO red cells and to normal red cells. What we found, however, very interestingly, is that those children that have SAO are much, much better at blocking the interaction between Duffy binding protein and the Duffy receptor. So while 22% of children with SAO had more than 90% blocking ability in an in vitro assay, only 6.5% of normal children, wild-type children, had the same amount of blocking. So SAO does not change Duffy itself, but somehow there is an interaction between the ability to acquire immunity to that interaction with the presence of that trait. So the conclusion from this little study here, and that brings me to almost the end of my presentation, is that SAO does offer very strong protection against vivax clinical disease, but it does not do anything against falciparum. So we have all this protection against vivax, but the only thing we find for falciparum is protection against cerebral malaria. And I think we can therefore postulate that vivax is at least likely to have contributed to either the selection or at least the maintenance of that polymorphism in the Pacific populations. And I quickly want to have just a little highlight what we are doing now. Where do we go from here now in terms of our epidemiological research? There's a lot of biological research still ongoing in these different cohorts. On one hand, we have started some work to look at we are starting some work to look at the comparative epidemiology of transmission. So we're starting to ask the question not just who is infected and who gets sick, but turning it around and looking at the whole issue from the mosquito perspective and says, who is infectious to me as a mosquito? So who transmits the two species? When is it transmitted? And how efficiently is it transmitted? We are starting to do work on comparative population genetics, and we're doing with Eliza Barry, also here at WEHI, we're doing some classical microsatellite population genetics work for both species. But we also have started collaboration with the Broad Institute and Sanger to start looking at that question on a whole genome level. We have conducted 2 different cohorts since these ones, where we have selectively removed liver stages from children by giving them treatment with primaquine, and are now looking at the differential risk of infection, disease, and transmission in children that have liver stages and children that have them removed. And we are also working together with Jim Kazura, who talked here very recently about visited WEHI, as part of the South Pacific Isomer project. We are looking at the effect of intensified control on these kinds of host vector parasite interactions that I've talked about. So I want just to finish with saying that all this work is obviously not mine. I'm the spokesperson here for a very, very large group of people at the P&G Institute of Medical Research, the Case Western Reserve University here at WEHI, as well as the Swiss Tropical Institute in Basel, the University of Western Australia. And as I mentioned before, most importantly, none of this work could have done without the unbelievable support we get from the Papua New Guinean people and the children and their guardians, as well as the staff at the different health facilities with which we work. Thank you very much.
Thanks, Ivo, for a really, I found an absolutely fascinating presentation. It's really overturned quite a few preconceptions. So questions, we'll start with Andy.
So is maternal antibody against vivax less abundant or less efficacious for falciparum?
Good question. If you look at the actual, now I've switched it off here. If you look at the actual timing of when you see, in particularly also infections in very young children, then you will see that in the very young children here, also for vivax, in the maternal antibodies will last up to about 6 months, and then they're gone. So you see really here in these very young children, you have very low levels of infections that you actually can detect by light microscopy. You have somewhat more infection by PCR, which probably means the maternal antibodies are there and they are able to suppress the vivax infections quite well to low densities. And therefore, they do get infected, but they do not establish high-density infections as long as they have the maternal antibodies. And then as the maternal antibody weans, you start seeing that you start getting more and more high-density infections. We are doing studies. We have a number of studies together, particularly with Chris King from Case Western, where we have looked at placental transfer of antibodies, and vivax antibodies are as efficiently transferred across the placenta as falciparum ones. We find also there's one thing that might be different. There is potentially slightly higher risk of congenital transmission of vivax malaria. The vivax parasite is much more flexible in its membrane. It can much easier squeeze through small gaps, and that may lead to a somewhat higher. There is some indication if you look at IgM in cord blood, for instance. There is more vivax IgM in cord blood than there is falciparum in cord blood. And also when you do PCR, there tends to be but the data is limited. But I think generally, it would be similar. We have just now an NIH grant with Chris King where we're actually going to start following a cohort of children through the first year of life, where we have followed the mothers during pregnancy, and you know what their exposure was during pregnancy in terms of falciparum and vivax exposure. And we're now following these children very detailed for the first year of life. So hopefully that will give us an answer to that. But at the moment, our indication is that it's not an issue of inefficient transfer of maternal antibodies.
Any other questions?
Why do you think that children with mixed vivax and falciparum infections have more life-threatening severe malaria? Do you think that the vivax and falciparum are independently driving severe disease?
I think that is probably the case. What tends to happen is these children tend to have a higher total parasite biomass, and that probably they tend to have, in that sense, more of the kind of toxins that are produced. The other thing we need to remember is that vivax is a much more efficient pyrogenic agent than falciparum is. Vivax will cause tremendously high fevers and very high cytokine, particularly TNF levels, at much lower parasite density than Plasmodium falciparum does. We do not know exactly what drives it. I mean, we would expect that the GPI of both parasites is so similar that they would target the same effector cells, but there clearly is vivax, and that may then, that strong pyrogenic effect of the vivax may contribute to the pathology by falciparum and make it more dangerous.
I thought your finding that the number of clones was really the best link to the severity of disease that was fascinating. Do you think that is a biomass issue, or more potential for trouble, or just a sign of less immunity?
Now, it's definitely not, in that cohort here, it's definitely not a sign of less immunity, because these kids have very little immunity. And we are really dealing here, in a sense, with uncomplicated disease, not with severe disease in this cohort. So they have very little immunity. Actually, the incidence, as you saw, the incidence of disease goes up as they get older, and if they would acquire significant immunity, it should be going down. The reason why it goes up, and it's studies we know from vector studies, is that the number of mosquito bites that you get as a child depends on your body surface. So a small baby gets bitten less often than a 3-year-old or a 4-year-old because it is smaller. And we see this here. We see that the number of new clones a child gets increases with age in parallel with the increase in disease. So it's really probably, that's the reason why we have that increase in disease in falciparum. And so there is not much immunity. I think it is so direct because you get, basically because the immunity is little, but not every clone. You also saw there's about twice as many clones, or 3 times as many clones as there is clinical disease. So not every falciparum clone you get will lead to disease. Some of them will just be asymptomatic and hang around and disappear. What the difference is, whether these different VAR genes expressed or it's a parasite that has already some immunity exists, these are questions which we can look at in more detail. But I think really it is simply a lottery thing. You get more new parasites that are genetically distinct, that have different antigens that they're exposed, that come in, more chance you have that one of them will find a gap in your host defense and will create severe disease, will create disease. And I think that's as simple as it is.